So good morning, everybody. I'd like to thank the organizers also on behalf of my co-authors for giving us the opportunity to share with us our data, not only in the poster 2128, but also during this oral session this morning. And I have nothing to disclose other that this work was supported by a grant from the Helmholtz Fund. So our starting point here is that, of course, the functional role of the medial prefrontal cortex has been intensively investigated, in particular in studies looking into decision-making, although of the last 10 years, we memory researchers have also made quite some progress in finding a function of the medial prefrontal cortex in various memory processes, actually. And just to give you some examples, this is somewhat older work from Atsuko Takashima, where we actually looked into the recognition of more recent and remote memories. Atsuko actually conducted a study that was consecuted across 90 days, and actually she got subjects to memorize stimuli, and over time, of course, the older stimuli became more and more remote, and she was very nicely able to show that there is a sort of a shift from a decrease in functional activation in the hippocampus, that is shown on the upper left side, to an increase in the functional activation over the dorsomedial prefrontal cortex as successful recognition of remote items came in. And later on, Jan Willem Thielen extended Atsuko's work in our lab, and he actually looked into the functional connectivity that was associated with this activation change, and he was basically able to show that within this interaction between the medial prefrontal cortex and the hippocampus, in particular the midline thalamic nuclei kick in during the first 24 hours of this consolidation process, and they kind of mediate this interaction between the medial prefrontal cortex and the hippocampus, so that information is actually stored in a reliable context, something that has also been supported very much, of course, by seminal animal literature of people like, for example, Thomas Süthoff. So, of course, functional MRI, event-related fMRI, offers us only indirect measures of neural activity, and given the advances we have also in other imaging facilities, the questions, of course, like how at the human system level can we start to investigating the neurotransmitters that are involved. From animal literature, we know that neural activation is related to GABAergic and, of course, excitatory, for most likely glutamatergic transmission, and that these induce different cellular processes resulting in long synaptic alterations, as, for example, LTP. But we have, of course, had quite some progress in MR spectroscopy, which allows us nowadays also not only to have these measure these broad NAA or Cholin peaks, but we can also use differentiation processes, for example, with megapress, that such that we can look in learning-related changes. For example, we've had quite some nice recent studies that have tried to dissociate the GABA concentration across a variety of learning tasks. It has been shown, for example, that learning novel motor skills decreases the GABA concentration in the motor cortex. Likewise, Michaels and colleagues have shown that there's, in fact, also, again, a decrease in GABA concentration after a working memory course. And finally, we've had some recent evidence that there's an increase in glutamine, so GLX concentration when subjects engaged in a mental imaging task, kind of reflecting the excitatory neurotransmission that is needed to process succession. So in the light of this aforementioned previous work, we were kind of interested whether these changes in the medial prefrontal neurotransmitter levels are actually associated with memory. So we know that we have these medial prefrontal activations, and we want to know whether we can relate these functional connectivity of this previously defined network also to changes in inhibitory and excitatory neurotransmitters. In order to approach this question, we set out a study where actually 27 subjects, out of which 18 were female, with an average age around 23 years, participated. And as you can see on the upper side, we actually conducted a single voxel spectroscopy at the medial prefrontal cortex, dorsal medial prefrontal cortex. I'll get to that in a second. And thereafter, we used actually a very classical associative memory paradigm, a phase-name association that has been shown to reveal a robust encoding activation. After this encoding of phase-name pairs, subjects underwent a second session of MR spectroscopy, and then outside the scanner, we would actually do a paper pencil recognition test so that we could get a subsequent sort of trials according to the subsequent memory performance, again as done by the previous speaker, into trials with high and low confidence. So with respect to the proton magnetic resonance spectroscopy spectra, we of course made use of MegaCRES, which actually induces sort of an interleaved data collection, which allows us with a sort of editing sequence, pulse sequence, to actually have a sort of a differentiation, a subtraction of the GABAergic and glutamatergic peaks from the more overlapping peaks, as for example N-acetyl aspartate and choline. So if you subtract these from each other, then actually you, in this added on section, you can actually quantify the GABA and the glutamine peaks. However, we have to be aware that often the GABA concentration is still somewhat lower than the glutamine peaks, and I think this is also something we need to keep in mind as we will look at our results in 1 minute. Just to share a little bit behavioral results with you, as this was, of course, this classical memory paradigm. So indeed, we had the high confidently remembered phase name associations. The memory performance was above chance level. However, the low confidently remembered items, the memory performance was at chance level. So we think that they are likely not associated with the recollection of specific episodic details, but rather guessing. And this is very much in line with more older behavioral memory literature we know before. And for the neural activation, so we are actually contrasting also high versus low subsequently remembered items, because this gives us this differential memory activation we are actually interested in. So if you look a little bit at the neuroimaging results, what we basically did is that the functional activation of the mean peaks of the fMRI voxel associated with the memory activation fall within the mean position of the MRS voxel, and therefore we use this cluster also as a seed region for our subsequent PPI analysis. And looking at the B results, B basically did not find a change of GABA between the first and the second spectroscopy scan. However, the glutamate concentration increased significantly over time, and I'm happy to discuss what this may mean. And most importantly, also with respect to our experimental question, we were able to show that this increase in the excitatory neurotransmitters correlated positively with memory performance. So we set out to dig a little bit further into the neuroimaging results, and this whole brain picture just is here to show you that the subsequent memory-related activation actually covered brain regions, including the hippocampus, the fusiform gyrus, and also the inferior frontal gyrus, very much in line with other studies of successful associative memory encoding. We then actually looked into the relationship between the functional connectivity analysis and also the glutamine, and this trial this picture kind of reflects our stepwise approach. So first of all, we investigated whether within this data set which kind of functional connectivity we would find, and so seeding within the medial prefrontal cortex and contrasting the high versus low confidently subsequently remembered trials, we were able to find an increased functional connectivity to the right hippocampus for this successful memory activation. And then we integrated, of course, the glutamate concentrations as a covariate of interest, and we found that the functional connectivity between this medial prefrontal and hippocampal network is related also to a change in glutamate concentration. Now we wanted to split this a bit further ahead, so we actually try to have a sort of a mediation kind of analysis where we actually integrated an indirect effect into this path, whereby next to that direct effect between glutamate being related to an increase in memory performance, we checked whether the functional connectivity between the medial prefrontal cortex and the hippocampus, which is shown on the A side, would actually mediate effect, or whether it would be more the functional network enclosing the midline thalamic nuclei, which is shown on the right side. And we basically found that the thalamic interaction did not was not significant, so this indirect effect is not a mediating effect. So to sum up, we explored the dynamics of the inhibitory and excitatory neurotransmitters and also their relationship to this functional connectivity in this medial prefrontal thalamo-hippocampal network and memory performance. And we found that actually the glutamate but not the GABA levels were increased as a function of episodic memory encoding, and that this was actually associated with a stronger functional connectivity. And we think that it is interesting to further pursue this work because we think that this neurotransmitter reactivity will help us to understand how this facilitates the formation of detailed and vivid memories. And I would like to thank you for your attention by this. Thank you.
All right, thank you. Unfortunately, we don't